Hi, my name is Sohail and today I am going to talk about composition of chromosomes. In this lecture, I will focus on nucleosomes and nucleosome string, histone, chromatin and chromosomes. Each chromosome is made up of DNA tightly coiled many times around proteins called histones that support its structure. Chromosomes are major carriers of genetic information in eukaryotes, and chromosomes are present inside the cell nucleus. Chromosomes means colored body and chromosomes virtually colorless. The term refers to their ability to be stained by certain dyes. Chromosomes are physical carriers of genes, and chromosomes are made of chromatin, and chromatin is composed of DNA and protein that is called histone. Genes are the reason for the existence of chromosome. Chromosome packaging is facilitated by certain proteins that is known as histone. Histones have a positive charge because they have high proportion of amino acid with basic side chains. The positively charged histones associate with DNA, which has negative charge because of its phosphate group, to form structures known as nucleosome. The fundamental unit of each nucleosome consists of a bead-like structure with 146 base pairs of DNA wrapped around a disc-shaped core of 8 histone molecules, 2 each of 4 different histone types. There are 5 major classes of histones, that is H1, H5, H2A, H2B, H3 and H4. Histones H2A, H2B, H3 and H4 are known as core histones, while H1 and H5 that is known as linker histones. Each nucleosome bead contains a set of 8 histone molecules forming a protein core around which the double-stranded DNA winds. The DNA surrounding the histone that consists of 146 nucleotide pairs, and another segment of DNA that consists of about 60 nucleotide pairs long links nucleosome beads that is known as linker DNA. So this is one nucleosome and this is second nucleosome. In between these two nucleosomes, this is a linker DNA, and linker DNA is made up of about 60 nucleotide pairs. Let's watch this 3D rotational model of nucleosome complex. The nucleosome was originally defined as a bead plus a DNA segment that links it to an adjacent bead. Today, the term more commonly refers only to the bead itself, that is 8 histone molecules and the DNA wrapped around them. Nucleosome function like tiny spools, preventing DNA from becoming tangled. The role of histone molecules is more than simply structural because their arrangement also affects the activity of DNA with which they are associated. Histones are increasingly viewed as an important part of regulation of gene expression. That is, genes are either turned on or turned off with the help of histones. The wrapping of DNA nucleosomes represents the first level of chromosome structure. So this is the first level of chromosome structure, and the higher order of structure of chromatin leading to the formation of condensed chromosome. So this is the higher order of chromosomal structure, and this is the first order of chromosomal structure. The diameter of DNA molecule is 2 nanometer. The nucleosomes themselves are 10 nanometer in diameter. The packed nucleosome state occurs when a fifth type of histone which is known as H1 histone, that is linker histone, associates with the linker DNA, packing adjacent nucleosomes together to form a compacted chromatin fiber, and its diameter is 30 nanometer. In extended chromatin, these fibers they form large coiled loop held together by scaffolding protein, non-histone proteins that help to maintain the chromosomal structure. The loops then interact to form condensed chromatin found in the chromosome, and cell biologists have identified a group of proteins collectively called condensin required for chromosome compaction. Condensin 1, condensin 2, condensins are large protein complexes that play a central role in chromosome assembly and segregation during mitosis and meiosis. Condensin binds to DNA and wraps it into coiled loops that are compacted into a chromosomal structure. Organization of a chromosome that include nucleosome, nucleosome string, chromatin fiber, supercoil and chromatids. After the replication of DNA, each DNA molecule that begins to coil around a histone core, and approximately 146 to 200 nucleotides of DNA, they wrap twice around the core of 8 histones, forming a nucleosome. One histone molecule is associated with a small segment of DNA which is called linker DNA, and this histone molecule is called linker histones. So this is a linker DNA which is present here between two nucleosomes. So the whole DNA is 2 nanometer thick and is turned into a chain of beads like appearance called nucleosome string, and its thickness is 10 nanometer. The nucleosome string begins to coil again about its axis, and it forms another thick fiber that is called chromatin fiber. Its thickness is around 30 nanometer. During G1 and G2 phases, that is gap 1 and gap 2 of cell cycle, chromosomes they are found in this level of organization. The chromatin fiber shows two regions, that is heterochromatin and euchromatin. The loosely packed region that is known as euchromatin, and the densely packed region that is known as heterochromatin. Heterochromatin is highly condensed and unexpressed region throughout the cell cycle, and transcription process does not take place in heterochromatin region. Euchromatin, that is non-condensed region, and the genes of this region are also expressed because the genes are active. So heterochromatin, which is highly condensed throughout the cell cycle and is generally inactive in transcription. And what is transcription? It is the synthesis of messenger RNA from DNA, that is called transcription. So the genes in heterochromatin region are not transcribed. Euchromatin, which is less condensed and contains actively transcribed genes. So the transcription process that takes place in euchromatin and transcription process does not take place in case of heterochromatin. When cell undergoes division, then euchromatin is also condensed so that a uniform chromatin fiber is established. So this is the region where supercoiling of chromatin fiber takes place, and its diameter is 300 nanometer. Supercoil establishes into a chromatid with diameter of 700 nanometer as a result of further coiling around itself. To sum up, chromosomes are major carriers of genetic information, and chromosomes are present inside the nucleus. Chromosomes are composed of DNA and protein that is called histone. Genes are the reasons for the existence of chromosomes. Histones are positively charged and the DNA is negatively charged. There are 5 major classes of histones, that is H1, H5, H2A, H2B, H3 and H4. H1 and H5 are known as linker histones, and rest they are known as core histones. The role of histone is important for gene expression as the genes are turned on and turned off, and DNA is packed with 8 histones to form a nucleosome. A nucleosome, its diameter is 10 nanometer. The beads of nucleosomes, it form a nucleosome string. The nucleosome string begins to coil to form chromatin fiber, and chromatin fiber has 2 regions, that is heterochromatin and euchromatin. Heterochromatin is highly condensed and the genes are never transcribed. Euchromatin is less condensed and have actively transcribed genes. Now the chromatin fibers, they supercoil to form a chromatid of 700 nanometer. Cell biologists have identified a group of proteins, that is condensin 1 and condensin 2, that is collectively known as condensin, required for chromosome compaction. Let's watch this slideshow.